in Kenya, we know that vaccine rollout has started. It started with the healthcare workers. Now it's going to the teachers, to the essential services, the security, and people who have comorbidities and who are older. And there's a lot of talk. People are asking about the effectiveness, the safety. Is it that it's a vaccine just for the African man? And today we have experts with us who will tell us about this vaccine and tell us what we need to know about the vaccine. We have Professor Omo Anzala, who's a senior research scientist at KAVI, Dr. Mary Beth Maritim, who's an infectious disease specialist. We also have Dr. Christabel Haemba, a pharmacovigilance at the Pharmacy and Poisons Board, and Mr. Edward Mumbo, who will be taking us through this very important discussion. So I'd like us to be able to start. For our presenters, we can start with you, Professor Omo Anzala. You can unmute and introduce yourself.
Yeah, am I able to share my screen? Hello?
Yes, you can share your screen.
Okay. So can you see my screen?
Yes, we can see your screen.
So do I start or I wait?
You can start.
Yeah, okay, thank you so much. And I'm glad we are here to talk about this. But what I'll do is to step a little bit back and also bring in the question, how new pathogens emerge. Because we might be talking of the current problem, but we have had problems in the past, and the emergence of new pathogens will actually continue. So the first thing here is that a lot of infections you see in the humans actually originate from the wildlife and the wild, the animals. And these are infections we call episodic cycles or zoonotics when they come to humans. And anytime you read literature and you hear a virus called a human virus, human herpes virus, human immunodeficiency virus, it means there's a counterpart of that one in the wild, and hence the word human. And quite a number of these viruses spilled over sometime in the past from the wild to the human because of the close nature we have with the wildlife. Either we are using wildlife for economy, we are using wildlife where we live, and these are how these infections move over from the wild into the humans. And definitely you have not seen the last of this. You'll keep seeing spillovers, and there are constant spillovers that maybe never reach epidemic or pandemic proportions. So the ones we normally see are actually the ones that reach epidemic or pandemic proportions, like, for example, the COVID, the SARS-CoV-2 COVID we are talking about. So emergence is now clear. And now, one of the things that is also clear that COVID is not the first documented SARS-CoV emergence into humans. 1889 to 1890, we had a pandemic. And recall, I'm not talking about the Russian, I'm not talking about the Spanish flu. This was called the Russian flu or the Asiatic flu in 1889. And this started in the Russian Empire infected quite a large population and also had mortality of that nature. Initially, it was thought to be caused by influenza virus. But with the modern advancement in medicine, the archived samples have been looked at, and it's clear that this particular outbreak then was actually caused by a coronavirus. And this is the coronavirus we currently call the OC43, which is now endemic worldwide. So why am I saying this? Because the reason I'm saying this is that what we are seeing is not new. And what we are going to see in future, you'll actually be able to see spillovers. And then when you look at coronaviruses, ladies and gentlemen, you'll find that there are 4 human coronaviruses. And the OC43 is actually here. It is spilled over in 1889 and now has become endemic. And definitely the 229E, the NL43, the HK-UI, all this definitely spilled over at some time from the animal side into the humans. Not long ago, 2012 to 2013, we had another emergent Middle East respiratory syncytial coronavirus. This again infected quite a number of people. It caused deaths. And what we know now is that this particular virus, SARS-CoV, is now endemic in the Middle East. We have been working with this virus among the camels, and quite a number of people are infected, although at a very low level. So this is now has again become a human coronavirus. In 2002, there was another outbreak of SARS-CoV, severe acute respiratory syndrome coronavirus. This happened in mainland China and Hong Kong, and it never went beyond this region. And again, it caused deaths as well as morbidity and mortality. Just showing you that these spillovers will continue to occur. But then what do we do as healthcare workers, and what do we do as regions and countries? And also to let you know that these outbreaks can occur anywhere because of our interaction with the wild. Now, currently now talking about SARS-CoV-2, we know that the current virus, the SARS-CoV-2, has telltales that it originated from a bat. This is the lineage transmission from a bat. And we say that because when you look at the bat coronavirus or the dog coronavirus, because they are there, or the camel coronavirus, when you sequence them, there are telltale features because of the kind of receptors they use to let you know that this is not from a bird and not from a human, maybe from another animal. So the latest data by the group that was sent to Wuhan, China, to find out the origin, it is clear that the origin of SARS-CoV-2 is from a bat. But it didn't move from a bat to a human directly. It moved from a bat to another intermediate host, which is another animal yet to be identified. And it is from this animal that it eventually got into the human. So this intermediate host is really what we want to know, because usually when these viruses are in the wild, they have certain receptors they use to infect, and certain what can I call surface proteins that are critical for binding to receptors. When they now move from the animal to an intermediate host, eventually to humans, that's how they begin to change, those particular molecules within the surface to be able to infect humans through binding to specific receptors in humans. So we now know the origin, but now the problem is identifying the intermediate host. And then coronaviruses, as I've told you, we have them in the wild. We have the human ones, the 4, and now we have 6, and SARS-CoV-2 is likely to now to be the 7th. We have coronaviruses in pigs. We have coronaviruses in dogs, in cats, in camels, in bats, in snakes, as well as in birds. And it is interesting when we talk about snakes, because when people think of snakes, they only think of the venom. But from where I see it, when you have a snake bite, I also think of a virus that a snake can transmit through that bite, even if you survive. So snakes do carry viruses, and especially the constrictors. So it is interesting that this ability of these viruses to move from from animals to humans actually exists. So that then makes you understand when you ask the question, where do these viral agents come from? Because the animal kingdom is actually full of these viruses. And now, coming closer to SARS-CoV-2, this is how the virus looks like. This is the cartoon of the virus. And these are the proteins. It is enveloped. It has a linear strand RNA, which is positive for those who are interested in understanding that. But really of interest to us is the spike protein, is the nuclear capsid protein, it is the membrane protein, it is this envelope protein, it is this single strand RNA. These proteins are all of interest, all of them, because each of them has a function in terms of infectivity, in terms of replication, and also in terms of causing disease. But at the same time, they also make targets for purposes of treatment, but also for purposes of vaccine development. And if you look here, this just shows you the genome structure from the 5' end, where you have where actually the ribosome binds to begin the processes of translation. So all of them are important, like the spike protein. 2 subunits, the S1 and the S2, very critical to binding on the angiotensin enzyme 2. And this is the initial step in which now the virus is used to enter into cells. And realize that once it enters, it binds onto the cell, the entire membrane is actually left out. What is injected into the cell is actually just the genome itself to begin the processes of translation. So it is good to understand that this is one of the critical proteins, but all these other proteins are also important in terms that they can be utilized as targets of treatment, but also targets of vaccines. And then on a micrograph, this is how the coronavirus looks like. When you look at the electron micrograph, and you can see different shapes, but what is critical is the spike proteins, which gives it this idea that it looks like a crown, and hence the name corona, actually meaning a crown, but then we add the virus itself. So that is actually the description. So it is important to notice this. And then there is one bullet here that is critical. And I will say it now, and I will visit it later, that coronaviruses have a high frequency of recombination during replication. And this is a point we should have noted, because people are worrying now about SARS-CoV-2 variants. But to us who have been in this field, it is not a surprise, because these viruses have a high frequency of recombination, and when they recombine, new viruses or new variants actually emerge. So that what goes or infects a human, when that virus comes out from that human, it is not exactly the same as a virus that caused infection. Because when it goes through the processes of immunity, the immune pressure selects those viruses that have escaped. So what comes out of the human is slightly different from what caused the infection because of recombination. And this aspect of the virus and the way it replicates, in the sense that when the genome, and it's only the genome that is released into the cytosol, is that it is ready to be translated into the various proteins. And it is this technology that is also critical and was used to make some of the vaccines, because here you have different messages, and each of these message is coding for a specific, is actually geared coding for a specific protein. Like, for example, the longest one is the polymerase protein. The next one is actually the hemagglutinin on the cell surface. The next one is actually the spike. The next one is the envelope. The next one is a membrane. The next one is actually the nucleocapsid. So you can actually see that the technology that was used to make the mRNA is actually more or less the same approach the virus uses to replicate itself. Although the genome comes in straight, the transcription before it is actually transcribed makes sure that it gives the ribosomes different messages, which then code for the different proteins. And then eventually these proteins come together in a very complicated assembly method, and eventually a virus is released. So it is actually the same method. Like now, whatever you see them saying, the message, this particular one, that particular message is a clear messenger RNA that is specific and coded for the spike protein. And this is the knowledge that is now available. And these are some of the knowledge that has always been there and was actually used in terms of actually making some 2 or 3 of the various platforms for the vaccine. And then let me not belabor this, because I've already said actually the replication cycle that it goes through, but it is more or less the same. The various things, assembly, and then the virus is released. Now, there is a lot of talk about variants, about which variants of concern, variants of interest. The classification of SARS-CoV is still growing. So you start with mutations, and several mutations make up a variant. And several variants make up one clade or one lineage. And then the virus has several clades and lineages, and then you go onto the virus itself. So really the point is that these variants and these mutations, how do they occur? And they occur because of what I've told you, that the virus is very prone to immune pressure and to recombination. And this is the way the classification in one of them, called the NextStrain Clades, where you see a hierarchy. Like the 19A was the original, maybe the Wuhan strain. And when it started spreading because of immune pressure in different population, from there, then we have various variants that are in 20A clade. And then those who are in 20A are slightly different from those ones who are in 19B, and slightly different from those ones which are in 19A. So when you hear about B something something, A something something, it means that that A is the clade and then the various mutations. For example, that will be a particular mutation, but you see the origin of the way it went. So the 20A, then they start mutating, and several variants, slightly different from 20A, then become 20B. So you can actually see that we have so many different clades and so many different variants, because each clade is made up of a variant. So this is, ladies and gentlemen, the elephant in the room. And these recombinations and clades have an issue. If you look at another aspect of taxonomy, these are the two. This is the NextStrain clade classification. This is another one we call the Pango lineage classification and others. So you can be able to actually realize that we are still dealing with a virus that we have not totally agreed on how to classify. But nonetheless, we can sequence. For example, you can actually see that for the two variants of concern, the UK one, the B117, and the South African, the B1351, for the Pango, they are in B. All right? But for the NextStrain, they are actually, one of them is in 20I, the other one is in 20H. But for the other ways of nomenclature, we are still very, very fluid. All right? So different ones will come there. The Brazilian one for the Pango is called a P1, but in the NextStrain, Clade is actually called the 20J501V3. So this area will actually grow, and we shall keep ourselves posted. And then coming closer to Kenya, the last time I checked, this could be about maybe 3 or 4 weeks old. But so far we had sequenced about 700 sequences. This one fell into 8 clades, and then in them we had 34 lineages. You can actually find that at that particular time, we had only, the B117 had only been identified from one sample. This is the UK strain. The B135, 351 had actually been isolated from 6 samples. But this data is about 4 weeks old. Things have actually changed. We are actually seeing a lot of the UK strain, and we are also seeing an emergence of the South African strain. What we have not seen yet the Brazilian strain. And these others are just the various variants we find within the country. So this is an area, again, that is growing. So I will now leave that at that, and then come closer to the various mutations. So mutations don't only occur in the spike protein. Mutations can actually occur in all the genes of the SARS-CoV-2 virus. So when we concentrate on the spike protein, then maybe sometimes we miss the point, because mutations elsewhere in the envelope, in the membrane, in the nuclear capsid can actually be actually have consequences of even the disease outbreak, because all of them have a role to play in terms of how cells are infected by the immune responses these particular proteins elicit once the virus is replicating in the host. Now, there are 2 aspects of new variants, currently circulating variants of concern, but there's also another term that has come in, variants of interest. These variants involve the spike gene mainly for now, but we are beginning to cast the net wider to look at mutations in other genes, as well. So a variant is a variant of concern if it escapes diagnosis. And this means that somebody presents with all the clinical features highly suggestive of COVID. But when a sample is taken, the sample comes out negative. Then it means that that particular variant in that particular person has escaped the diagnostic feature. And that will mean that the problem is not in the spike gene, because we don't use the spike gene for diagnosis. We use other genes. A mutation, a variant can be of importance when it undergoes vaccine escape. And this is the concern that is going on now. A variant will also be important if it causes more infections, if it's more infectious. A variant will also be, or a variant of concern, if it has severe outcome, either more mortality and more people to be hospitalized. That is what we call a variant of concern. And then there's a variant of interest. A variant of interest are those variants with multiple mutations, but we don't have epidemiological history attached to them. Currently, they are talking of 2 individuals whose samples were sequenced from Tanzania, and they have found multiple mutations with these variants, but we don't have any clinical data or epidemiological history related to them. So for now, we cannot call them variants of concern. We call them variants of interest as we investigate. And then moving now closer, why are vaccines made? Vaccines are made because of is a public health tool. And I'd like us just to visit the natural history of infectious agents. This is how it happens. One is susceptible and becomes infectious. Or one is susceptible, becomes infectious, they are treated, and once they are cured, they become susceptible again. The first scenario is HIV. You are susceptible. Once you get infected, you are infectious and infected for life. So the natural history of HIV fits this first scenario. This second scenario of susceptible, infectious, susceptible actually fit other sexually transmitted infections. And these are like gonorrhea, mycoplasma. These are infections where you become susceptible, you become infectious, you are treated, you become susceptible again. So these are the infectious agents we call non-immunizing infectious agents. Somehow, because of the way they are in their nature, the body is never able to actually mount any form of lasting immunity. So HIV falls here, and on this one, other STIs, gonorrhea and the like. Then there's another very interesting scenario here, where you become susceptible, you get infected, you recover, and once you recover, you develop immunity, and you develop immunity for life. And then a second scenario here where you are susceptible, you are infectious, you recover, you develop some immunity, but you become susceptible again. So the first group are diseases we know, measles and rubella. These are immunizing infectious agents, but very different outcome. The second one is like influenza. And now we are seeing that SARS-CoV-2 is actually falling among this second scenario, where you recover, you become immune for a while, but because that virus in us is in a state of drift, it is changing. Your immune system only covered you for some initial variant. The new variants are not amenable to the immune system. Then again, you become susceptible, all right? Or your immunity is not long lasting. It wanes over time. Again, you become susceptible. So these scenarios are critical in terms of understanding this, to ask yourself, are we capable of developing an infection? So you just remember these ones: susceptible, infectious, recovered, immune, totally, and those that you keep going round and round. Although they are immunizing, this is classical. That's the best we would like to have. But this is where we are with SARS-CoV, and that's why, for me, I tell all of you, prepare for the long haul. SARS-CoV-2 might end up like influenza, where we shall keep having surges every so often as our immunity wanes, and at the same time, the new variants come on board. And how are vaccines made? Really, the infectious agent must be understood. We must also understand correlates of protection and correlates of cure. And these come actually from the natural history. From here, we know quite a number of patients are exposed, all right? For our population, a majority of people recover, and there are those majority of people who actually, even when exposed, never develop disease. So that exposure, infection, and outcome is actually critical in terms of understanding correlates of cure and correlates of protection. So that has now been understood. And then once you do that and you know the correlates of protection, then you know the virus, then you understand that really you want to develop an antigen based on the correlate of protection. And the correlates of protection now we know are two binding antibodies and neutralizing antibodies specific to the spike protein. And the aim here is to elicit an immune response at innate immunity as well as adaptive immunity. And the adaptive immunity, you want to ensure you have memory cells that will give you cytotoxic T cells and memory B cells that will actually give you plasma membrane and plasma cell and eventually the antibodies, which will bind and neutralize or bind, and the virus will be engulfed. So this is the initial understanding of how then the correlates of protection lead you now to the vaccine targets. And for now, the target that has been chosen by nearly all the front runners is a spike protein, because this is the one that has shown that is a correlate of protection. Those who survive develop large amounts of antibodies against the spike protein. And again, the animal studies that were done showed clearly that antibodies against the spike protein actually protect either from infection or from disease. But all these other targets can actually also be targets of vaccines. As we know, the live attenuated vaccines or inactivated vaccines will actually give you immune responses to all these targets. And then these are the various platforms, and Mary Beth will talk about one or two or three of these vaccines, but the various platforms we have, you can have DNA vaccines, you can have RNA vaccines, and you can have the live attenuated vaccines, you can have inactivated, you can have protein, and then these are the new platforms, the viral vector and the viral vector, either replicating or non-replicating. The current vaccine we are using in Kenya now, AstraZeneca, is a viral vector, but non-replicating. And that should actually be very clear. These others are there. So I will not go into details in this, but the interest is that once you've picked your gene of interest, now the spike protein, this is the protein that you'll either use to insert into your vector, all right, into your, the genome of the vector. And then now this becomes your virus particle of interest. When this particle infects and replicates, the gene of the vector replicates, but also this spike protein is replicated and comes out as a protein, which then elicits the immune response. But nonetheless, all these other platforms are on the way, and with time, we shall be able to talk about them. And then the components of the vaccine, because when you talk about adverse reactions, the adverse reactions will be caused by any of this: the antigens, the adjuvants, the antibiotics, the preservatives, and the stabilizers. So these are actually the components of the vaccine, depending on how the vaccine is actually made. So when we talk about adverse events following immunization, we want to find out, are they because of the antigen? Are they because of adjuvants? Are they because of the antibiotics that are put in there to avoid the vial being inactivated, actually infected by bacteria? Or are they just preservatives and stabilizers that we are reacting to? So it is also good to know that these are actually standard components. They will vary from vaccine to vaccine. And then the clinical trials. From phase 1, once the antigen has been made, to phase 2, to phase 3, and then submission, and then eventually phase 4 introduction. So a lot of this work for SARS-CoV-2 started around May, June, July for phase 1s and phase 2. So basically, a lot of individuals have gone into this phase 1, phase 2. So we have fairly sufficient data to say that the vaccine was safe through phase 1. We have sufficient data to say that the vaccine was also safe and immunogenic in phase 2. And currently, we have both emergency use authorization going on and at the same time, the clinical trials for phase 3 are still ongoing. So sufficient data is actually coming in to tell us the issues of efficacy and also to tell us the issues of side effects. So we are confident enough that whatever vaccines we have are safe enough and they have an efficacious level. What we now want to learn is how do we move from efficacy to effectiveness? And those are some of the discussions that Mary Beth will actually take us through. So part of the current front runners is a Johnson & Johnson, which is under emergency use authorization. We also have the AstraZeneca Oxford vaccine, it's a viral vector. This is the one we have. We also have a protein subunit, all right? These are also being used in some countries, especially in Mexico. And then we have another Pfizer biotech, which is an mRNA. And then we have this particular one, the Sputnik, which is a viral vector. The AstraZeneca carries a chimpanzee adenovirus. The Johnson & Johnson carries a viral vector we call adenovirus 26. And then the Sputnik carries two adenoviruses, AD26 and AD5. And then this particular one, I think is from China, it is whole viral inactivated. So, and then you can see the various participants who have been involved in various of these clinical trials. So out there, there's actually sufficient data on immunogenicity as well as efficacy. The critical thing now will be the effectiveness. And then viruses, vaccines either protect from disease or they protect you from infection. So my last slide will be here just to say that we actually want a vaccine to protect us from infection. If it can't protect us from infection, we get infected. We want the vaccine to prevent us from getting disease. Or if we get disease, then we want it to prevent us from getting severe disease. And this should actually be the stop. If not, then we want it to prevent us actually from death. But at the end of the day, we also want that vaccine, as it protects us from this, we want also it to protect us from transmission to others. So that even if I have COVID, I don't transmit to other people. So I will stop here and thank you very much for listening.
Thank you very much, Professor Anzala, for that detailed presentation. You've indeed taken us through the basic of vaccinology and even how all this came about, and we really appreciate it. Thank you, our participants, also for continuing with us. And you can post in your question and comments at the Q&A section, and you'll be able to get to them at the end of the sessions. Right now, I want to get into another session and welcome Dr. Maritim. Dr. Maritim, you're about to take us through the next session. You can put up your slide. Thank you very much.
Evening, let me share my screen. So good evening, everybody, and thank you for this opportunity to make this presentation. And I'll be focusing on vaccine safety and adverse events following immunization. Just give me a minute. I'm trying to share the link with some people who are trying to log in. Just give me 1 second.
Dr. Melibeth, it won't help. We are already.
Oh, we are full. Oh, yes. I feel so bad. Okay. Okay. So I'll focus on COVID-19 vaccine safety and adverse events following immunization. And basically, this is the outline of my presentation. We'll talk about Covishield specifically. So a little bit about the vaccine components, adverse event following immunization, safety and safety monitoring, and then a little bit about vaccine efficacy. because many people want to know if the vaccine that they are taking will really protect them. So we'll focus mainly on Covishield because this is a vaccine that we are rolling out, and this is a vaccine developed in partnership between AstraZeneca and Oxford University. And I think Prof. Anzala had this slide where he was talking about the vaccine construct. So for the Covishield vaccine, which is ChAdOx, it has a vector, which is a chimpanzee adenovirus. And that vector carries genetic material that will encode the spike protein that basically is a coding for the spike protein. And this was developed because already the initial studies suggested that the spike protein stimulates and is the antigen that stimulates an antibody response. And thereby this has been used to design the vaccines. So the vector is modified, it's non-replicating, it doesn't, it's not able to replicate beyond the first multiplication within the host. It's unable to cause disease because it's from the chimpanzee, it's an adenovirus from that would affect the chimpanzees. And basically what happens when you inject the vaccine, the cells express the spike protein and the body is able to produce antibodies against the spike proteins. And these antibodies are protective against any infection, external infection by SARS-CoV-2. So basically, to summarize, we have a viral vector that is non-replicating, but it carries the genetic material that enables our body to form antibodies against the spike protein. And once following the vaccination. So in terms of the components of the vaccine, these are just in general. The vaccine would have active ingredient. In this case, it would be the vector that's carrying the genetic material encoding the spike protein. And then it would have adjuvants. In the case of the AstraZeneca vaccine, it doesn't really have any adjuvant. It may have residual trace elements, which are substances as a result of the manufacturing process. So in some of the vaccines, not necessarily in the COVID vaccines, some may have formaldehyde as a residual trace elements. And then some vaccines have preservatives and stabilizers because some of them have a short shelf life, some of them have a fairly long half-life, and they have to move from different destinations to reach the destination of use. And therefore, they would require some preservatives and stabilizers. So for example, in the ChAdOx vaccine, polysorbate 80, which I'll talk about in my next slide, is one of the stabilizers. And then water. The vaccine already comes reconstituted in water. So water would be one of the ingredients. So the importance of knowing these different components is that in understanding adverse events following immunization, it can occur following contributions by these different components that would be in various vaccines. So for the ChAdOx vaccine specifically, it has the active ingredient, which is the vector encoding with the spike protein. And then it has its excipients or other things that are mixed with it. So it has histidine, L-histidine, hydrochloride, monohydrate, polysorbate 80, which is a stabilizer. It has sucrose, ethanol, monosodium, sodium and magnesium chloride, EDTA, disodium, and water for injection, which is the vehicle that I've talked about. So in terms of, many of you have seen this slide before. It's almost like my favorite slide. I cannot present without having this slide. But basically, just to re-emphasize, what is an adverse event following immunization? So it can be a symptom, a sign, a lab finding, or a disease. And this occurs after immunization without necessarily having a causal relationship. So it may not really be caused by the immunization process, but any symptom, sign, lab finding, or disease that occurs after the immunization then would be described as an adverse event following immunization. And there's a committee where I sit in, and it does what is called causality assessment or adjudication to see there's a causal link. So a causal link is where you can actually say from science or from evidence or from what we know about how the vaccine is designed, you can actually say the vaccine caused that adverse event. So in the broader definition, anything or any of these characteristics that comes after then would qualify as an adverse event following immunization and needs to be reported, and then adjudication is done thereafter to see if there's a causal relationship with the immunization. And this brings me to this also slide, one of my favorite slides. In the vaccine world, we have 5 types of adverse events following immunization. Type 1, which is vaccine product related reaction. And this is an adverse event that is caused or precipitated by a vaccine due to one of or more of the inherent properties of the vaccine product. So it's attributable to the vaccine product. And that's why I had that slide that was talking about what would you consider in the bottle, for example, that has the vaccine. So it may be related to one of the ingredients, so to say, that is within the vaccine vial. Then type 2 is vaccine quality defect related reaction. And these are adverse events that are caused by or precipitated by a vaccine that is due to one or more quality defects of the vaccine product, including its administration device as provided by the manufacturer. So you're looking at maybe a failure in the manufacturing process or a defective equipment to administer the vaccine that is part of that vaccine, because some vaccines may require a special device to administer or to inoculate the vaccine on a specific area. So that would be classified as vaccine quality defect related reaction. One common example that we use in this group of vaccine is, for example, when we are talking about the process for some of the live attenuated vaccines, where you have a whole organism that's supposed to be passed through some process to make it less effective, to attenuate its activity. And when you give the vaccine, it can revert to virulence. So that is an example of a quality defect, whereby during the manufacturing process, if you did not fully inactivate the vaccine, and then it causes an adverse event of that type, then you'd say vaccine quality. So it's about the manufacturing process and the quality related to that. Type 3 is the commonest type of adverse event following immunization. And yet again, it's the one that is amenable to correction, because when you train the healthcare workers, you can actually minimize this. And this is immunization error related reaction. And these are adverse events that are caused by inappropriate vaccine handling, prescribing, or administration. So if you don't store the vaccine well, it might become contaminated. If you don't administer it well, you might injure a nerve. So these are the types that you'd call immunization error related reaction. Type 4 is immunization anxiety related reactions. And these are adverse events that arise from anxiety about the immunization process. And we see this typically for some of the vaccines that are administered to adolescents, when they're all sitting on a bench, queuing, waiting for the vaccine, and then they can start to faint. So one faints, another faints, another faints, but it may just be related to anxiety about receiving the vaccination and the fact that they are seeing the person ahead of them receiving the injection. So it can create anxiety. And then coincidental events, which is type 5. These are adverse events that are caused by something other than the vaccine product, immunization error, or immunization anxiety. So this last group is really a classification by exclusion, that it did not fit here, it did not fit here, it did not fit here, it did not fit here. And therefore you're saying it's coincidental. And sometimes some events start off as coincidental, but as newer knowledge emerges, then you might find actually that they may be classified into different other groups as more information is generated going forward. So having classified or described broadly the types of adverse events following immunization, when we receive a vaccine, there are those side effects that we expect because of the vaccination process. And some of these have already been defined at the phase of clinical trials, because when we conduct clinical trials, and Prof. Nzala mentioned this, we go through different phases, assessing different aspects of the vaccine. So we mainly classify vaccine reactions or side effects into either minor or severe reactions. So minor would occur within a few hours of injection. They resolve after a short period of time and pose little danger. They may include local reactions like pain, swelling, or redness at the injection site. But also we may have some systemic symptoms like fever, malaise, muscle pain, headache, and loss of appetite. Then there may be serious or not serious, severe reactions. But this classification is more at the point of like clinical trials when there's a sponsor and they can classify what you would consider as minor and what they may consider as severe reactions. So in terms of distinguishing minor and severe, this usually do not result in long-term problems. They may be disabling, but not permanently. They are rarely life-threatening. They may include seizures, allergic reactions caused by the body's reaction component of the vaccine. So these are the two broad categories. And going back to explain the mechanism of what we are saying minor and what to expect. When we give the vaccine, we want it to induce an immune response. So we should not be surprised when we get some of the side effects, because it shows us that the vaccine is actually doing what it's supposed to do. So at the point of injection, you can have what is local reactions at that point, but you can also have systemic effects. So in the local reaction, basically what mediates that is this production of cytokines or chemicals around that place that are part of the immune response to that vaccination. And this leads to symptoms of swelling, pain, and redness. So when we receive our injection, we should not be surprised when we feel pain on that arm, when we have swelling and redness. So this is a local reaction because of stimulation of the immune response locally. Some of the chemicals or cytokines that are produced can be carried in the bloodstream and reach distant organs, and therefore mediate the systemic effect. So they go into the blood, they're carried in the blood, and they cause effects away from the site of injection. And some of these side effects that are mediated through this process include headache, fever, and fatigue. This illustration again explains the different pathways. So we have an immediate or a fast neural route, which is mediated through the nervous system. And then we also have a slow route that's mediated through the bloodstream. So in the nervous system, our nerves in the surrounding area are able to transmit the painful stimuli to the brain, and it's perceived as pain. Then you have other influenza-like symptoms and other processes that help us to be able to feel or mediate or perceive the reactions. But the reactions that are transmitted through the bloodstream may take a few days to occur because the chemicals are transported to distant organs like the liver, and then the liver is able to mediate the immune response that is now generalized and carried out in the rest of the body. So this is just a summary, and we should not be surprised after receiving the vaccine when you get headache, fever, tiredness. Those are just expected side effects. So in terms of looking at the two reactions, remember we had talked about the minor and then the severe. But in this slide, I just want to focus on what we would call serious reactions, which are of interest to us when we sit in the safety committee, because these have to be reported and they have to be acted on immediately. So what would be a serious adverse event following immunization? So this is any untoward medical occurrence that results in death, requires inpatient hospitalization, results in persistent or significant disability, is life-threatening, and in pregnant women, we also add if it results in congenital malformation. So that is an additional of what you'd consider as serious reactions for which you have to put in place an intervention to investigate and assess and see whether there's a relationship with the vaccine, to really classify in one of the 5 types and try to assess if there's any causal link that may be related to the vaccine. This is just an illustration to tell us about frequency and the different classifications of different adverse events that occur and how we rate them. So you'd say an event is very common if it occurs very frequently, so more than 10 of a particular population, followed by common. So common is between 1% and less than 10%. Uncommon is anything less than 0.01%, less than 1%. Rare is less than 0.01% to less than 0.1%, and then very rare, of which the blood clots would fall in this category. so less than 0.01%. So looking at the various classifications. So this is in a broad sense, and when clinical trials are conducted, you have to classify and rate based on the frequency of occurrence within the study population. And one of the things we have to understand is that sometimes the numbers in clinical trials may not be large enough to pick some of the very rare or fairly rare events. And that's why when you deploy the vaccine, for example, if a vaccine has already attained market license, then you should have post-marketing surveillance. In the case of COVID-19 vaccines, they've been deployed under emergency use authorization and none has attained market authorization. So one of the requirements under emergency use authorization is that we are also doing safety monitoring to try and identify some of the rarer side effects that may not have been seen with the limited numbers within the clinical trials. And we are talking of currently close to 300,000 doses or people have been vaccinated globally. And that gives a large number of people to be able to study some of the rare or to be able to pick up some of the that may initially start signals, but now as we get more data, then may be actually attributed or associated with different vaccines. Profanzala probably had this slide as well, but I just wanted to highlight for the ChAdOx vaccine, which is the third one here, we have the polysorbate 80, which is one of the stabilizers. For the other mRNA vaccines, we have the polyethylene glycol for the Pfizer vaccine and Moderna vaccine. All these are stabilizers, and they have been reports of anaphylactic reactions, particularly with the Pfizer vaccine, which have been assessed and may be attributed to presence of polyethylene glycol, which is the molecule that actually encapsulates the mRNA as part of the delivery to stabilize it as it's being delivered intracellularly. The equivalent of that in the ChAdOx vaccine is polysorbate, but we haven't had cases of anaphylaxis or fatal anaphylaxis related to the ChAdOx vaccine. Sorry, we've already gone through this slide, so I don't have to have it here. So coming back to the vaccine and the data from the clinical trials, what were the rates of the different adverse events? So that even when you're vaccinated and you find that you have headache, you should not be too worried about that headache because it has already been documented as part of the very common side effects. So again, very common, occurring in 1 in 10, more than 1 in 10. So headache, nausea, fatigue, myalgia, malaise, pyrexia, chills, injection site related side effects. So tenderness, pain, warmth, redness, itchiness, swelling, and bruising. So darkening or change of color around that specific area. The common side effects are occurring in more than 1 in 100. So in that group, we have vomiting. And then uncommon means it's fairly rare. So occurring in 1 in 1,000 individuals. So decreased appetite, lymphadenopathy, specifically over the area on the same arm where the injection is given, dizziness, abdominal pain, excessive sweating, body itchiness, as well as rash. So this is the proportion of the different side effects that were noted in clinical trials. And even within our data and safety monitoring within the PPB portal, the distribution is fairly similar to what has been seen globally. So a little bit about anaphylaxis. So it's a very rare but severe, potentially fatal reaction. And it would occur minutes to 1 hour after getting the dose of the vaccine. And one of the safety nets to minimize the risk of anaphylaxis is that when you receive your vaccine, you have to be observed for 15 to 30 minutes within the facility before you go away, so that any anaphylactic reaction can be managed before it progresses. So what are some of the signs of severe allergic reaction? You may present with difficulty in breathing, swelling of your face and throat, tachycardia, having a rash all over your body, dizziness and weakness. So this is what observation basically looks out for to be able to identify anyone who may develop anaphylactic reaction. So if someone develops anaphylaxis to any components of the vaccine, if one is known to have had maybe a prior episode of anaphylaxis to any of the components that I had shown in an earlier slide, which are contained in the AstraZeneca vaccine, then the vaccine would be a contraindication. You cannot receive the available vaccine. If someone develops an anaphylactic reaction following the first dose, then we do not give them the second dose because it's predictable. Once you have that predisposition, it's fairly predictable that you are likely to develop another anaphylactic reaction following a second dose, as opposed to some reactions that are called idiosyncratic, that are not predictable, are not dose dependent, and may not even be replicated following a repeat dose of the same vaccine. So for the ingredients, because they are predictable and anaphylaxis is predictable, you don't give someone with predisposition to anaphylaxis the vaccine. This slide is just supposed to highlight to us the numbers in clinical trials and to just reassure us that in these vaccines that are being deployed, they went through all the clinical trials. And when we read the supporting literature that was used to apply for emergency use listing or approval with WHO, you'll actually find very detailed information about even animal studies, the studies in rats, where they were looking at effects on fertility. And you actually find a lot of information, some of it which is so very detailed, but it's important in the era of transparency that you can be able to actually to access that information and review it and be able to see that whatever was done is sort of in a transparent manner. So in phase 1 clinical trials, you're looking mainly at safety and immunogenicity with fairly few numbers. In phase 2 clinical trials, you're looking at safety, side effects, immune response, and also dose scheduling, so determining the optimal dose. And again, fewer numbers. In phase 3, you're looking at efficacy. So you do phase 3 clinical trials to determine how efficacious the vaccine is in a controlled environment, and we'll talk about that towards the end. But you're also generating data on safety in a larger, a fairly larger population. And then phase 3B, where we currently are under deployment, when you're deploying these vaccines under emergency use authorization, we are still generating a lot more data on safety, but also looking at effectiveness in the real world, which may be lower than efficacy in clinical trials, because in clinical trials, it's within a controlled environment. So these are just some of the numbers that were involved in the various clinical trials: 50,000 for AstraZeneca, 60,000 for J&J, and different levels, different numbers for the different clinical trials. And the importance of knowing this is that when you're looking at or analyzing the safety data, we may say that 50,000 maybe gave us some of the safety data, and therefore some things that may not have been seen because they were fairly rare may be seen now when you give the vaccine to many more people. So, and then again, looking at the efficacy is also important. Although the total number maybe has no bearing in terms of efficacy, some of the things that can affect efficacy is the time when the clinical trials were conducted. If you conduct clinical trials when maybe cases are going down, you might overestimate the efficacy of a vaccine compared to if you give the vaccine when cases are going up. So I just have a few slides looking at thrombocytopenia and coagulation disorders. This is what it has been probably trending, and this is talking about the clots. So just to simplify about the clots. So a combination of thrombosis, which are clots, and thrombocytopenia, in some cases accompanied by bleeding, has been observed very rarely following vaccination with Vaxzevria. This is a vaccine, the Chaddox vaccine, that's being used in the UK and over the European Union, which is similar. So when you talk about the vaccines were designed, but they're different manufacturers. So manufacturers were given a seed lot, which now they use to make additional vaccine. So our vaccine is called Covishield, but they share a lot of properties with this vaccine. So the events currently that have been reported have been within the European Union jurisdiction. And this information I got from the EMA website, as well as even the insert for that specific vaccine, which has actually since been updated to incorporate this new safety concern that has been described. So the patients presented with clotting, they had low platelets, and in some cases they were even bleeding. And this, the parts where the clots were noted were in very unusual sites. And these were in the brain. We have what is called the superior sagittal sinus and other sinuses. So cerebral venous sinus, there were clots within the sinus. There were also clots within the abdominal veins, what are called the mesenteric vein thrombosis. And then there were clots within the arterial. I haven't gotten much clinical data to see which anatomical areas were affected with the arterial thrombosis, but there's been reported thrombosis within the arterial system. And in these cases, this was accompanied by what is called thrombocytopenia, where you have reduction in your platelets. So majority of these cases occurred within 7 to 14 days following vaccination, with most of the cases occurring in women under 55 years of age. However, in those settings, a lot of women have presented themselves for vaccination. So again, there might be an overrepresentation of women, but we are also talking of cases so far for the cerebrovenous thrombosis, a report of 163 cases. For the mesenteric vein thrombosis, a report of about 53 cases out of 25 million doses of vaccines administered. So we are talking of 25 million cases of vaccine to identify about 250 events. So giving a ratio of, I'm just trying to calculate the ratio of one in about 2.5 million. So to identify one case, you probably need to vaccinate 2.5 million people. Some of these cases had a fatal outcome. About 18 of the cases lost their lives. So healthcare professionals should be alert to the signs and symptoms of thromboembolism and thrombocytopenia. And basically in the patient presentation, some of the patients presented with shortness of breath, chest pain, leg swelling, and persistent abdominal pain, which followed vaccination. And we've talked about within 7 days to 20 days following vaccination. For the ones who had the venous sinus thrombosis, they presented with severe and persistent headache, blurred vision, so they couldn't see clearly, or they had, some of them had bruising of the skin beyond the site of so the reason why I'm emphasizing this, remember I talked about headache as a side effect that you'd expect. But if it's severe and it's persistent, then that would be a worrying headache, which may be one of the symptoms you would be concerned about as maybe manifesting the cerebrovenous thrombosis. Blurred vision, again, is when you can't see clearly, and then bruising. Remember I talked about bruising, but if the bruising is away from the site where you've received the vaccine, then perhaps that would be something to be concerned about. So having said all this, I came across this publication done by Dr. Onyambu with Prof. Amayo and Dr. Kitonyi, and they were looking at clinical features and patterns of imaging in cerebral venous sinus thrombosis at KNH, because many of us may think these clots are rare, but this is a publication that was done in the East African Medical Journal in 2013. So in terms of the study design, this was a retrospective study design over a 5-year period. So the study was looking at chart reviews of patients admitted at KNH over a 5-year period between 2006 and September 2011. And the main aim of the study was to describe the clinical characteristics and patterns of neuroimaging findings in patients with radiologically confirmed cerebrovenous sinus thrombosis. So that clot was already written about in 2006 and was studied in Kenyatta. So we should not think that this clot will be coming for the first time because of COVID-19. It's already been described within our setting. I may not have access, perhaps there is more newer literature looking at this, but again, I found this very, very interesting because the patient population that was studied and had the findings may not be too different from the patient population within the European setting. So in this 5-year period, there were 51 patients who are already confirmed. The interest was to describe the radiological findings. So they already had confirmed cerebrovenous sinus thrombosis. And because there were 51 patients over a 5-year period, it means we are looking at perhaps about 10 per year. So in terms of the study findings, The median age of the patients was 30 years. The commonest age group was 25 to 34 years. There were 38 females, so majority of the patients were females, and they had clinical presentation of headache, seizure, neck pains, neurological deficit, visual disturbances. And then in terms of risk factors in this population, 39, more than close to 40% of them had infections as a risk factor for developing the cerebral venous sinus thrombosis. 13% of them, close to 14%, had just delivered or were in the period after delivery. So having a procoagulant, like being in a period where they were at risk of thrombosis. And then just about 4% were were taking oral contraceptives. So what this shows us is that cerebral venous sinus thrombosis is rare, but we have had it it's already been described within our setting. And this information is useful for us, particularly in the safety committee, because we'll need to look at what we call the background rates to see, for example, if you have the vaccine contributing to increase rates, then you might find that you're actually documenting much more cases. But one of the things I have to say is that this is just a one setting, one study site setting, and you may find actually maybe it's more described in other settings, but we'll be looking out for additional literature, maybe gray literature, to see and help us to establish even what is the background rate, even as we continue to roll out the vaccine. Because in terms of assessing causality, we'll have to make consideration for a lot of more other things going forward. So this is just a pictorial still from the paper, looking at the age distribution. So common again in the age group 25 to 35 years, but more, you can see the age group from 17 years to around 49, almost reproductive age group, again being the highest period where the cases were reported. In terms of clinical presentation of the patients, majority of the patients presented with headache, and we talked about headache that is persistent, very severe. Some patients presented with convulsion, some patients presented with neurological weakness, all the way different manifestation ranging all the way, with a few others who are not described in terms of what were these manifestations. So I just wanted to use that to illustrate that although we are talking about clots in Europe, these clots are not only confined to Europe. We've had a description of these clots even within our setting before even COVID was administered, so that people may not be deterred and thinking that only the vaccine can cause clots. So in terms of safety monitoring, I'm almost coming to the end of this presentation. PPB has developed an electronic reporting system where patients who develop adverse events following immunization can actually report through this portal and can access. So this portal is open for healthcare workers to report. It's also open for the general public to report, to access this and report any adverse event following immunization. So this is just a cycle of the surveillance process. So we have, if someone has an AEFI, it's reported, and then it's detected, all the data is collated at the PPB, and they develop a line list. So that's the notification, reporting. There's a process of investigating to gather more evidence, so that even at the time when you're doing causality, we have a lot of data that we are looking at to see if there's a causal link or other explanations that can explain the adverse events. There's analysis, and then the causality assessment process. And following that, there'll be feedback. So it's a continuous cycle that's called the surveillance cycle. And we've already put in place a lot of the mechanisms we deploy the vaccine. So in terms of the in-country reporting and processing of AEFI, I think, again, this is just assessing all that. The adverse events are reported from all over the country through a passive surveillance system. And this is collated at the level of the Pharmacy and Poisons Board through the electronic reporting system that I've talked about. They generate a line list. Then you have a lot of process investigations and getting a lot of data. So reporting, investigation form, case, clinical case report, getting lab reports, autopsy reports in case of loss of life, so that can be able to get all the data to be able to comprehensively investigate. Then this data is shared to global, provided to the safety committee to be able to assess the causality and be able to provide and complete the cycle that I've talked about previously. So in terms of the causality assessment process, when we're looking at trying to attribute causality or to classify, basically this is the ultimate process to try and classify all the adverse events following immunization into one of the 5 categories. So we go through this process where you ask these several questions and provide answers. So is there evidence for any other causes? Is there a known causal association with a vaccine or vaccination? So you look at the published literature, if there's any biological plausibility, did the test demonstrate the causal role of the vaccine? Could the vaccine have a quality defect? Could there have been an immunization error? Could the event have been immunization triggered stress response? So remember, this was type 4, this was type 3, this was type 2. And then for type 1 and for type 1 particularly, you want to assess if it was related to the vaccine component. So you're looking whether there is biological plausibility, and sometimes you have to do additional tests. So for example, with the issue of the clots, we now think that there could be a relationship because of the signals and the cases. So we need to find out where we can find a specific test. And the literature, some of the teams in Europe have already looked and demonstrated that there could be a platelet factor 4 antibodies that are generated in certain groups of people. So to also try and define who is really at risk. Is it an idiosyncratic a certain group of people, or is there a genetic predisposition? And what would be the clinical marker in those group of people, maybe even at the time when they develop the adverse event, to try and link that biological plausibility. And then if yes to 2, did the event occur within a plausible time window after vaccine administration? Is there strong evidence against a causal association? And then look at other qualifying factors to help classifying, for example, previous history of a similar event, looking at the background rate within the population of the event, pre-existing, present, and past health conditions within the person, potential risk factors and other medications, and then triggering factors. So this is a continuous process. And as it is now, we may not have a lot of literature, but when we look back later, when you've accumulated a lot of the literature, we might even find that maybe it's a specific substance within the vaccine or the vaccine's construct that is responsible for that. But as it is now, we don't have a lot of evidence in terms of saying it's not related to the vaccine or it's really related to the vaccine. So I'm coming almost to the end of my presentation. In efficacy and effectiveness, because many people are wondering, should I even take the vaccine? Will it really help me? So when you talk about efficacy, we're looking at the degree to which the vaccine prevents disease and possibly transmission within an ideal environment, so in clinical trials. And you compare a vaccinated group and placebo group and see how many people in the vaccinated group got COVID and how many in the placebo group got COVID, and then you compare. But one of the things we have to remember, even when you receive the vaccine, it doesn't work overnight. You need a couple of days for you to mount an immune response. And most of these vaccines are most effective when you've already received your second dose. So it means even within the 2 groups, you might still have infections. But if a vaccine is working well, you expect that the number of infections in the vaccinated group will be much lower than within the placebo group. In the real world, when we deployed the vaccines like we currently are doing, we talk of vaccine effectiveness, which basically refers to how well the vaccine is performing in the real world. So some of the vaccines that we currently have, like Moderna and Pfizer, they have fairly high efficacies, 94.5% for Moderna, which are mRNA vaccines, and 90% for Pfizer. So the challenge with some of these mRNA vaccines, they require a lot of infrastructure, which we don't have in our part of the world. And therefore, they would be difficult to deploy because you need fridges and freezers to maintain very cold temperature. So because of the high efficacy, you also expect that these vaccines would have high effectiveness in the real world. And particularly for the Pfizer vaccine, this has been demonstrated. It has a very high effectiveness when they've deployed it in Israel, where they've vaccinated a large number of the patients And from the real world data, the effectiveness was actually close to 90%, with clinical data efficacy of 90%. So no difference between effectiveness and efficacy. In terms of our vaccine, AstraZeneca vaccine, it has an efficacy of 63% against symptomatic infection. And this is data from the clinical trials, which are done in different settings. So the UK, Brazil, South Africa. And this was when you have received 2 standard doses. In this data, what they found out is that from day 22 after receiving the first dose, there was no vaccinated person who was hospitalized, compared to 14 people who are hospitalized from the unvaccinated group. So that was able to generate the efficacy data. However, additional studies that have been recently published have shown that the efficacy of the vaccine is higher when you space the first dose and the second dose over a period of close to 12 weeks. So I know many of us who received our vaccine, we are actually going back for our second dose within 8 weeks. But if we delayed by 4 more weeks, we might actually have a higher efficacy. The immune response following that boost of the second dose is much higher with a longer interval. What we also know about our AstraZeneca vaccine is that it seems to have a reduced efficacy against the variants of concern that Prof. Nzala has mentioned, the UK variant and the South African variant. But one of the things we have to remember, so this is about symptomatic disease. For all the COVID vaccines we currently have, all of them are 100%, have a 100% efficacy in preventing the most serious outcomes that we want to achieve. They prevent severe COVID, critical COVID, and even death. So if we think 63% is too low, many of us are concerned that even if we were to get COVID, we don't want to be sick enough to go to the ICU, because it will cost us a lot of resources to be in the ICU. And we even know that currently we don't have ICU beds or oxygen, a lot of oxygen within our setting. So I think those are really good benefits if you're looking at 100% efficacy in terms of reducing the most serious outcomes that we are trying to prevent. So we had this slide, Prof. Nzala and I have had it, but I think it was just to highlight the numbers of the clinical trials. And then again, this one is showing the different efficacy. So these are the efficacies in the clinical trials, but in terms of severe diseases, all this then converge at 100% for the different kinds of vaccine. So for the most important outcomes that we really want to prevent, all the vaccines are the same. So thank you so much for your time, and we'll be able to take questions. Thank you.
Thank you very much, Dr. Maritim, for that informative presentation. Indeed, we are learning a lot in today's session, and you've covered quite a number of important things that are important concerns that people are asking about safety, about the efficacy level, and about even the effects of the vaccine. And we really appreciate that. We'll still call you back to keep reminding us. And thank you for honoring us with your presence in this webinars. For now, allow us to go into our third presentation. We are privileged to have Dr. Christabel Kaemba, from Head of Pharmacovigilance from the Pharmacy and Poisons Board. Dr. Christabel, karibu.
Thank you. Thank you, Agnes. So just to continue from where Dr. Maritim has stopped. So today I'll just focus on the reporting of AEFIs in Kenya. And this is just a flow chart showing how reporting is done. And as you can see, sorry, let me just put the slideshow. Yeah, so reporting, as you can see, starts all the way from the caregiver and the patient. So in the routine normal vaccine program that we've been having in the country before COVID vaccine, we've been having 2 channels of reporting. We have the manual reporting, where we have people using paper reporting, and this one, the patient brings to the attention of the healthcare worker in case they experience any AEFI. And this is then escalated from the healthcare worker to the sub-county MOH or public health nurse. And then they'll escalate this to the county director or epilogistician. And then finally, this is submitted to National Vaccines Program, either directly or as aggregate data through the DHIS. And as you can see, we are told we are expected to report all serious adverse events within 24 hours. And then the other remaining AEFI reports within 7 days. But then we also have the PPB online reporting system, which allows you to send reports real time. And here the caregiver can also report directly to PPB. We can also have the healthcare worker reporting directly to PPB. And at between PPB and National Vaccines Immunization Program, we have the National Vaccines Advisory Safety Committee Secretariat, which prepares the line lists that are submitted to the National Vaccines Advisory Committee for evaluation and causality assessment. So the vaccines program also prepares a joint, submits a report to the WHO through the joint reporting form, where they report the total number of people vaccinated and also the adverse events occurring. And through PPB, we are members of the WHO International Drug Monitoring Program, that is the National Pharmacovigilance Center. And the data we collect, we also share it with the WHO, and it goes into the global database that is for individual case safety report, referred to as VGBase. The market authorization holders and any sponsors of clinical trials are also expected to report to PPB. And at the end of the day, data is expected to be reviewed. And finally, we have the dotted arrows, which are showing the feedback that is expected after reporting. So in terms of COVID vaccine AEFI reporting, we are emphasizing that all adverse events following immunization should be reported. And for COVID-19, we are encouraging reporting through the online system because we require that this information reaches us immediately. And for those who've been vaccinated through the Chanjo platform also, once you've been observed for the 15 minutes, should there occur any AEFI, it will be reported in the Chanjo platform, but eventually this data will still find its way to the PPB platform. So this is how the EFI form looks like. And it's the same one that is on the online platform for healthcare workers. And as I said, you're expected to report all AEFIs, and key critical information that needs to be picked includes the brand of the brand name of the vaccine, the manufacturer, the dose number, if it's the first or second dose, and then the batch number. And for those who've been vaccinated and have already received the information or through text from the Chanjo system, you realize that the information has been containing the batch number and the name of the vaccine, and when you're expected to take your second dose, because all this information is key in during causality assessment. And we also need to know who is the reporter. So we need an identifiable reporter and also an identifiable vaccinee. And for reporting, you don't necessarily have to put the patient name, you can put the initials. And even if you put the patient name, when the data is being reviewed between PPB and vaccines program, it is decoded, and so the patient identifiers will not be there. And just an emphasis again, should you get an AEFI, you need to inform your supervisor immediately. If it's a serious AEFI, you manage the patient, and then within 24 hours, the same should have been reported to the platform. So this is how the online reporting platform looks like, what Dr. Maritim had said, and I'm going to do just a quick presentation of it when I'm done here. So far, just in terms of reported AEFI, so globally, by the time we're preparing this, about 6 million doses of vaccines, slightly over 6 million doses of the vaccines had been administered globally. And as at the end of March 2021, a total of 358,988 individual case safety reports on AEFI vaccines had been submitted officially to the global database for ICSR. And in Kenya, this is data that we had as up to 6th of March. As you can see, a total of 340,000 had been vaccinated. As of today, I think almost 500. And out of this, what has officially been reported to the PPB system is 279 AEFI reports, of which 272 were reported either as mild or moderate and having resolved or resolving within a short period. And the 7 EFI reports, these ones the reporters have been contacted for more information. They are under investigation, and soon the NVSAC will be able to give guidance on the way forward. So just to share with you globally, if you remember Dr. Maritim's slide, where she showed us what was expected in terms of AstraZeneca vaccine, what was seen during clinical trials. As you can see, headache has commonly been reported. This is the global database from all the countries that have so far been able to submit their reports. We have pyrexia, we have fatigue as the third most common. And coming back now to our local data at PPB, this is the picture of what we are seeing. And as you can see, it is resonating with what is being reported globally, with headache being the leading. And then we have fever, we have fatigue, we have myalgia, and we have generalized body malaise. So all these are reports that are just resonating. And these are just the top that reported, and we are still looking at the data, and more updates will come in as we continue interacting. So in terms of counties also, this is just to show us that, yeah, these reports are not from one county, they are coming from different counties, but we know there are other counties that started vaccinate, that probably have already sent their reports to the Chanjo system, and these will also be updated. So it's important that we report, because this helps us get vital data that will help in just strengthening the monitoring of vaccine. And then it enhances confidence to the immunization program, because the moment you report and you tell people this is what is expected, even the people will know when I'm vaccinated, this is what I'm likely to experience. And we'll be also reporting will enhance available local data. As you realize, most of the data that is being referred to is global data. And then early detection of risks will also enable corrective actions, and especially if there's an increase in frequency of the expected AD reactions, then we'll be able to know what information to give the vaccinees. And we are saying, if you're unsure of whether to report, just be proactive and then report the adverse events. So if you allow me, I'll stop sharing this, then I'll briefly share how the online platform looks like and how you go about the reporting. Yeah, so this is just a minute. Sorry, I hope you can see my screen. Yeah, so this is the online reporting portal. So it is, the URL is pv.pharmacyboardkenya.org. So if you come in as a member of the public, you just need to come and then click on this icon, and it will give you a report that looks like this. So for the members of the public, we need to say who is reporting. And this is important because a follow-up will be done, and we might need to get more information on the person who's reporting, the county where they're reporting from, your mobile number, because this is important in doing a follow-up, and then also an email address. And then you need to put the person who is reporting. Remember, we said we need to identify who is the vaccinee, and then the date of birth, or if you don't know, you can put the age categories. And it's important also to click and say if it's male or female. So you'll come here and you select the type of report you want to report. In case you want to report an adverse drug reaction, when you click on this, this is what will come. So to make it easier for the members of the public, we have selected some of the commonly reported side effects that cut across both medicines and vaccine. But should it be that it's not among this list, you can still come and type in the events. And then when did this reaction start? So the date, the time, the month, and the year. And also, is it still continuing by the time you're reporting? And then this one will just give information on the details of whether the medicine, the vaccine that you're suspecting caused the reaction. In case you want to report on suspected quality problems of the vaccine or medicine, when you click that, it gives you this information where you're able to say if you're suspecting something unusual in the vaccine, or maybe it smells weird if it's medicine. For vaccines, definitely the member of public will not go with it at home, but then they'll be able to report this if they are medicines. So in case you're a healthcare worker, we encourage healthcare workers to first register. So what it requires you is to create a username, a password, and then your name, your email address, and then you click if you're a physician, you're a nurse, or you're a pharmacist, you're a medical officer, or you're a lawyer, and then which institution are you reporting from? And then the code will automatically appear if you know the MFL code. If you don't know, it's not mandatory, and then counting. And we just want to know if the person typing in is actually human and it's not a robot, or it's not a computer generated thing. That's why we ask this question. So once you have done your registration, when you come back later, you can just come in and log in. So when you log in as a healthcare worker, so I'll log in and just demo how the report will appear. Sorry. So like now, when you log in as a healthcare worker, all the future reports that you submit, you'll be getting them on this dashboard. So the yellow form is for reporting suspected adverse drug reaction, that is from medicines. The white one is for reporting adverse events following immunization. This pink one is used for reporting suspected poor quality medicinal products, that is vaccines, devices, or any medicines. And then the green one is for reporting medical devices, because we've had cases where people have also had reactions or incidences as a result of medical devices. Like maybe you're using an auto-disabled syringe and accidentally you prick the person. So those are incidences that need to be reported. And then in terms of medication errors, you use this blue one to report. In terms of blood transfusion reactions, we use this, it's off-white form, that you use for reporting blood transfusion and any transfusion reaction. So for AEFI form, once you click on the form, and when you're reporting, you'll be able to get a generated that goes to your email. So here, in case you have any unsubmitted reports, you can always come back and then you'll be able to submit. So like this one, it's a report. I created this account and I was doing a demo on 25th. And as you can see, it is showing that I created this report, but I have not yet submitted it. So I'll just click on it to show you how it appears. So this is how the form will appear. And you'll need to put the name of the institution. So like if you start typing, it can give you some of the facilities that we have MFL codes, and you'll be able to pick them. And then like these facilities in Nairobi County, and this is the institution code, because these are already pre-filled. And then this is an initial report. You can always come back and do a follow-up report. So the sub-county, again, if you tick the sub-county, it will show you the sub-county that you're picking, the name of the patient. And as I said, you can choose to put initials of the patient. You don't have to put the name. And then date of birth. If you don't know the date of birth, you have the option of choosing the age in months. And this is a mandatory field. So it's important that you enter the same. And then the name of the guardian, if it's a patient, if it's a child, and then the phone number and all that, the village, and especially for vaccines, why it's important to do even village, because the issues of clusters, it could be that these reports are occurring in one particular cluster. And then the type of vaccination, is it static, is it mass, or is it an outreach? So like for COVID vaccine, it's static because people are going to a certain place and then they are being given. And then gender, you can also choose. So here, you've been given a list of the possible AEFIs that you can pick, but again, you can still click others. And if you click on others, you'll need to specify. Then you need to say the date the AEFI starts and the time. So when it comes to AEFI monitoring, it's very important that we know the time when the AEFI started, because this is used in causality assessment, and then you need to describe how the event occurred. So you can type and you can add as much detail as you have, and also the brief details on the event. So here, when you want to select the suspected vaccine, you just click this button and you'll be able to select. So like, for example, if you want to report COVID-19 vaccine, Covishield, you'll just click on that. And then the dose number, if it's the first or the second dose, and then you need to select the date. So like, let's say, assuming I've been vaccinated today and at this time, so it will pick the date and the time, route of vaccination, the site, the batch number, and manufacturer's detail. So you can add as many vaccines as you want, the ones that you're suspected. And then past medication history is important, because remember, as Dr. Maritim said, some of these adverse events could be coincidental. So we need as much information as we can. And in case you click that this is a serious report, then you'll need to say, why are you classifying it as serious? Is it because of a death? Is it because it was life-threatening? All that information. And what action did you take? Did you give any medication? Did you do any treatment? That's what you need to put. Did you collect any specimen? You're able to type in that. And then what is the outcome of the AEFI? Did they recover? Was it recovered with sequelae? Was it fatal, or is it unknown? And then if you have a pictorial or a picture just to show the reaction, you can click on that, and then you'll be able to choose from your folder and attach a photo and then give a description. So we get some times where you find that maybe a nurse has reported and then given the pharmacist to submit the report. And that's why we have these two, whereby the person reporting will put the details. And if the person submitting the report to PPB is different from the person who reported, if you click yes, then you'll be able to type in. But if you click no, it becomes idle and you're not able to. So supposing you're not so sure of the reports that you have done, you can save the changes and then come back later and do the report. Once you submit the report, you're not able to edit, but you can be able to download the PDF and put it in the patient file or put it in your folder. But when you go to your email, you'll get a unique number that will be generated once you've submitted the report. You can always go back to the link that will be on your email, or once you come here, you'll just come and click on the reports as I had shown you. And one other feature that you're trying to work on, and just to help going forward, you'll be able to get like more data and summaries. And this is the advantage of you being able to send in your report as a healthcare worker, because you'll be able to see summaries of what is being reported per county, per month. And we are trying to add more data in terms of type of AEFIs or type of ADRs that have been reported. Thank you so much. And I'll end my presentation there.
Thank you very much, Dr. Kaimba, for that. And it has indeed answered the questions in regards to pharmacovigilance and how to report the same. And thank you, our participants, for the questions. We'll be able to cluster some of the most frequently asked questions maybe towards the end. For now, allow us to go to a presentation by Mr. Edward Mumbo, all the way from Kwale County. They've been doing a lot of work there in regards to vaccine and immunization services, and we'll be hearing from him. Mr. Mumbo, welcome.
Thank you very much. Thank you very much. Reading the previous presenters, I'm not going to share any slide, but I simply want to talk about the targets that we are targeting for the vaccination this time round, and how the reporting is, and then also I want to discuss briefly about the uptake of the vaccine as it is today. And, probably take a minute to encourage people on why we need to come out to get vaccinated. So, like has been said, that the vaccine is very safe and very effective, and all of us need to come out vaccinated as expected by the ministry. We are doing this in 3 phases. Like the first phase started the month of March and is running up to the end of June. And this phase is very critical because it's the one that we target to vaccinate as many healthcare workers as possible. And when we talk about healthcare workers, we are talking about both the clinical staff and the support staff. All these persons that work in the hospital environment are today targeted for the vaccine. And then we are also looking at vaccinating teachers and also persons that are in the teaching institution environment. Therefore, in this regard, we are looking at vaccinating both teaching and non-teaching staff from educational institution or teaching institutions. We are also at this first time looking at vaccinating all the uniformed officers, and this we are talking about the army, the Kenya police, the prison officers, the KWS, the forest rangers, and all these persons that are categorized as uniformed individuals. We are also looking at vaccinating instructors in the religious institutions among the persons targeted in this first round. Fortunately, because of the morbidity and the mortality that is being witnessed in the country, recently the ministry and the government decided that we need to broaden the spectrum of the person that we are targeting for the vaccination to also target the persons that are 58 years and above. So those are the persons that we are targeting to vaccinate between now up to the end of June, not forgetting the persons that probably work in the migration department, based on the fact that when they are at the border posts, they're probably in the airport, in service units, they are bound to meet quite a number of persons from inside the country and from other countries, therefore broadens their exposure or their vulnerability. Therefore, these are the persons that we are really looking at vaccinating in the first round. The second round, we are looking at people with comorbidity, then people working in the hospitality industry, and the one working in the tourism industry. While the third phase will look like we want to vaccinate quite a number of the persons living in congested environments, like the ones working in prisons, detention camps, people working in probably care homes, people staying in the streets, and also these are other places that are overpopulated, especially informal settlements. So these are the persons that we are targeting vaccinate between now up to probably the end of 2023. And as at now, I want to say that the really primary target for the vaccination are the healthcare workers. But unfortunately, of the numbers that so far we have vaccinated from the time that the vaccine was launched in the country, we have hardly reached out to 50% of the primary target, which is probably the healthcare workers, the teachers, and the uniformed officers. Majority of the persons that are being vaccinated today are the cohort that was added recently, the over 58 years and above. So this is very unfortunate because as healthcare workers, we are very much high at risk. As teachers, we are very much high at risk. And also the uniformed officers are also very much high at risk. But there has been a little of a laxity and hesitancy among these cohorts that are primarily targeted for the vaccination. And reason probably is because of the fear of the unknown. And the majority of them, when you ask, they say, I will be vaccinated. But when they're going to be vaccinated, they don't tell you. So probably it was because of inadequate information that they have had. Now that we have had over 1,000 persons registered in this webinar, I believe and I want to be reassured that at least many healthcare workers have followed through the facts and figures of the COVAX vaccine and the benefits, and therefore we should be able to stand counted among the persons that have been vaccinated because the vaccine is very effective and very safe. Therefore, as healthcare workers, we have got 3 roles that we need to play critically. One, as a healthcare worker, as a teacher, as a police officer, we have 3 roles. One, we are the primary beneficiaries of the vaccine. 2, we are also community representatives in this matter. Therefore, if we are vaccinated and we go out there with a positive regard concerning the vaccine, we are bound to have as many persons as possible targeted to come out and be vaccinated. Again, we are advocates because if we are healthcare workers and we have been vaccinated and we know the facts and figures about the vaccine, therefore also we stand to serve as advocates for people to understand the significance of the vaccine and again also have them accept and come out vaccinated, because if the workforce in the health sector is sick, then we are not going to have anybody that probably will take care of the other sick individuals in society. Therefore, we need really to be our sisters and the brothers keeper in this regard, that we get vaccinated and also go out and also have others vaccinated. The Kenyan population, as it were, indicates that we are 50-50 in terms of gender. But looking at the facts and figures as from the facilities that have visited and the data that I've perused, there are more men vaccinated than women. The reason why probably they are fearing, or rather they are hesitant to be vaccinated, they are looking at talking about the safety of the vaccine. Now, from the previous speakers' presentations, I think we are all reassured that the vaccine is safe. Whether you are a lady or you are a man, we equally stand to benefit from the vaccine. Therefore, much probably we will discuss as we are addressing the questions and the concerns of the persons that have participated in this webinar today. Therefore, mine is to welcome us all to be counted among the numbers, because the benefits outweigh the risks that people are talking about in the country about the vaccine and her outcome. Therefore, thank you so very much for allowing me to talk to you in this webinar. And I believe that probably the talk that we have had from 7 o'clock up to now has touched our soul. And the soul that has been touched will also touch another soul, so that we see really higher numbers of the primary targets come to be vaccinated. Thank you very much.
Thank you very much, Mr. Mumbo. For now, we are coming towards the end of our webinar today, and we really thank our participants for staying with us. For now, we'll have Dr. Maritim. Dr. Maritim, if you'll be able to just do an overview of the common questions that have been asked and just give pointers as we even wind up.
I think my parting shot, I've seen a lot of frequently asked questions about whether the vaccine can be given to diabetics, hypertensives, patients who have had cancer. And the answer to that is actually those are the target groups, because those people who have comorbidities, people who have advanced age, more than 58 years, people with diabetes, hypertension, obesity, recovering from cancer, maybe underlying HIV, they're the people who, when they get infected by SARS-CoV-2 and they get COVID-19, they progress very fast to severe disease and they can die. So the aim of the vaccine is to prevent death and hospitalization, and those are actually the target groups for the vaccination. So the answer is yes, you can come and receive the vaccine if you have any underlying comorbidity.
Thank you very much. And as we've indeed had, the purpose of the vaccine is all the various vaccines that we have, they've been shown to have the 100% efficacy against serious outcomes. Professor Anzala, please will give us your parting shot also.
Say that again.
You'll give us your parting shot also for this session.
Yeah. No, for me, really, the whole issue of vaccines, we went through why vaccines are made and is the best public health tool you can ever have. and the other non-pharmaceutical preventive measures must be in place, and vaccine is one of the best additions. So really, for me, we should not be sitting in hospitals and waiting for patients to get sick. We actually must be out there looking for the very, very best solutions that will work upstream. Now we've talked about masking, we've talked about sanitation, we've talked about distancing, avoiding spreader events. Now that we have a vaccine, really that will be the best next way of ensuring that we stop our hospitals from being overwhelmed. So for me, really, we are the healthcare workers. We must be the front line to educate all of us that vaccines are actually made to work. We hear all these things. Some of them are because all these myths, issues of infertility, and these are stories, ladies and gentlemen. You've heard them before. They talked about polio, they talked about tetanus, and you can hear it is the same story being recycled. So surely we are better than that. It's the same story, infertility, it will cause this. All those are the same thing being recycled. So the truth is not there. The truth is that this is a global problem. We have a solution now. Let's embrace these solutions and avoid ourselves getting infected, our relatives getting infected, or our hospitals getting overwhelmed. Over.
Thank you very much, Prof. and Dr. Maritim and our team for really demystifying this information about the vaccine. And I hope for our participants we've been able to get the information, the right information that we need, and majority of our concerns have actually been answered in the presentations. And we'll continue to have this conversation as long as our COVID is here with us. We'll continue bringing you this information and equipping you with the right information for you to make an informed decision. So for now, we'll have to come to an end. Thank you for joining us. Do have a lovely evening. Thank you, everyone. Good night.
Asante sana, and bye-bye, everyone, for joining. We appreciate it.
Bye-bye.
Bye.
Right.